Hey everyone, I'm going to go through information on energy and enzymes. A lot of what is covered in the introduction of bioenergetics is information that we covered in our biochemistry section. So they talked about chemical reactions and that there's two different kinds. There might be endergonic or exergonic, which are the same as catabolic and anabolic, and describing whether energy is utilized for that reaction to occur or it's released by the end of that reaction. So those are some pieces that you should refamiliarize yourself with. But for the rest of chapter 5, we're going to focus on enzymes and what they do, what they look like, and how do they do what they do. So the first thing, what is an enzyme? We're going to be talking about proteins again. So remember that proteins go through and they have their 3D structure, and depending on what R groups are on the outside of that protein, that will give it its certain traits. So what pH does it like? What environment does it like? What's it going to interact with? That sort of thing. And if anything changes with that 3D structure, therefore its properties and functions will change along with it. Right? So those are enzymes as a quick recap for our proteins. What do they do? Enzymes are super important because they are involved in our metabolic reactions or in metabolism, aka they're involved in chemical reactions. So anytime a chemical reaction occurs, typically there's an enzyme involved with it. And for us as humans, there could be multiple enzymes acting in just one instance. Think digestion, right? How do enzymes speed up metabolic reactions? They go through and they're going to focus on what's known as activation energy, which I know was in our previous Edpuzzle video, but activation energy is going to be involved in any chemical reaction. So anytime a chemical reaction occurs, there has to be some energy at the beginning to get it started, to get those substrates moving or get the reactants moving around, right? So that's activation energy. Enzymes are going to have a lower activation energy compared to the regular amount, which would be higher, right? So in order to start the chemical reaction, it only has to reach this point rather than getting all the way up here with our activation energy. So that's why once it hits that peak, the chemical reaction occurs, and it goes a lot faster than having to wait to get all the way up to here. So again, here's the graph that goes along with it, right? Every chemical reaction will have some form of activation energy. We can see it's going to be a lot higher without, lower with, and we can see that change in energy with the products. So that'll drop down once the reactants go through their chemical reaction. So again, here's a recap of catabolic versus anabolic, or you could say endergonic and exergonic if you want to. Remember that catabolic reactions are going to release energy when they're breaking down their complex of molecules. So think digestion and breaking down food. This would be exergonic because they're releasing energy. Anabolic is going to consume energy or utilize energy in order to build molecules. So when we're trying to make something larger like a biomolecule or one of our macromolecules, that would be an anabolic reaction. Think endergonic for that one. And so we can see linking together amino acids to form muscle proteins. Here's another look at anabolic, utilizing energy, mixing with the molecules, in the end, creating something large so that energy gets stored up into the chemical bonds that's happening in our in our polymer that is formed here. Catabolic reactions are going to take this large molecule and reverse it, right? Break it down, release some energy, exergonic, have smaller molecules in the end. So like I said, enzymes are going to be those globular 3D proteins that have all bonded together. They are a form of a catalyst. So catalysts are anything that are going to change the rate of reaction. Enzymes are going to be organic compounds or organic forms of catalysts, meaning they have carbon in them, and they're allowed to increase the rate of the reaction. So anytime an enzyme is functioning, it's going to find its substrate or it's going to find its reaction and they're going to mix and link together. And that moment when they're linked together is called an enzyme substrate complex. Again, a reminder, substrate is kind of synonymous with the word reactant, but a little bit different because enzymes are just momentarily fixed with their substrates and they aren't there throughout the entire chemical reaction, or they aren't included in that. So enzymes are going to have a specific shape and a specific fit to them. So we kind of call it a lock and key model, or it could be called an induced fit model as well, where they're going to have a specific shape to them that's going to match up perfectly with a wonderful substrate specifically. Not all enzymes act on all substrates. So how do they interact with their substrates? They have an area or a region on that enzyme called the active site. So it's basically that zone that has a specific shape to it where the enzyme can come and lock into place. That's called the active site. And again, once they come together and join forces, it's called the enzyme substrate complex, where they have some weak bonds holding it together when the chemical reaction occurs. And then that enzyme quickly converts the original substrates that attached in there to the products, and then it releases, and then it's available for further use with other substrates that are hanging out there, and it can reconnect, create the complex, create the products, release. New one can go to that very same enzyme if it's available. So again, here's a look at that. There are a couple different types. Like I said, there's lock and key, where there's a perfect shape for every substrate, and there's something called induced fit. So we can see that this is a square, rectangular shape, and up here we have some globular substrates. So this would be an example of induced fit, where once these substrates come to the active site on the enzyme changes shape just ever so slightly so that these substrates fit in nice and neat to each other, and then they release out their products in the end. If it was a lock and key model, these would be, if this was rectangular like this, our substrates would also be in that rectangular shape and they'd have that perfect fit. So there are a couple different types. Here's our enzyme substrate complex again, when they momentarily latch on together and they have some weak bonds, and then the product leaves, and then the enzyme is available to bond with furthermore substrates later on. What affects enzyme activity? So by now you've done your POGL, which talked a lot about predicting the overall effects if you change the pH, if you change the temperature, right? So those things hopefully are ringing a bell. As I mentioned, the 3D shape is a key component for the enzyme, and if that 3D shape gets changed or altered, then the overall function of that enzyme is also going to be altered. So every enzyme is going to have an optimal pH and an optimal temperature range, right? So we're going to often see graphs that show us rate of reaction on the y-axis and then something else on the x-axis, whether it's pH or temperature or enzyme concentration, that sort of thing. Every enzyme is going to have its optimal range, and so you're often going to see those bell curves involved in those graphs that will show you where the highest peak is for the rate of reaction. That's where it works the best. If an enzyme is exposed to a range outside of its optimal range, right? So we're talking pH. If it is too acidic or too basic compared to what it normally operates at, then that causes the 3D shape of the enzyme to change. It almost like melts the enzyme, so that its active site is no longer available, or it's no longer recognized by that substrate. So they come towards each other and they're like, no, I cannot lock in and connect with you. I must not be the right enzyme. That's because that 3D change is called denaturing. Whenever there's an extreme temperature, an extreme pH, or outside of its range, it's going to change the shape and therefore change the function of the protein or change the function of the enzyme so that it no longer can interact. So we have some low grade temperatures that might happen, and that's good to get rid of certain bacteria or whatever else is in the area that doesn't really belong. Low grade is okay, and the enzymes can still sort of function. They're not quite at the um, apex of the bell curve, but they're close in that top range, so they could still function. But once we hit high grade fevers as humans, that usually pushes outside the boundaries of a lot of the enzymes that function for us. And so then that causes 3D changes and kind of a melting piece for our enzymes so that their active sites are no longer in the correct shape that they should be and they no longer can function. So that's where like digestion may not happen. Um, you might throw up, or you might have to use the bathroom quite often when you're sick because the proteins in your body your enzymes in your body are not functioning properly for digestion. So any food that goes in must come out because there's nothing that can break it down properly. Um, right, food poisoning, that sort of thing. So here's those bell curves that I was talking about, and a couple of them that you already looked at in your POGL over here, talking about pepsin versus, well, I guess we didn't talk about trypsin, but we're talking about stomach enzymes versus intestinal or pancreatic enzymes. So very similar to each other in their pH ranges. Okay, and then we can also see temperature between bacteria versus humans. So, and this was also listed in your POGL talking about PCR, right? Bacterial enzymes are utilized for PCR or whenever you need to make a whole lot of replicas of your genetic material. So anyone who's gotten a COVID test, whether it's a spit test or a swab or whatever, scientists will use, or lab assistants, whatever, will utilize PCR to make replicas of your genetic material using PCR, but bacteria works at a high heat optimal range over here. And during PCR, you do a series of like extreme temperature shocks and then freezing shocks to like increase the amount of genetic material that's involved. I'm going off on a tangent. Either way, let's just look at this graph and say, hey, humans operate at around 37 degrees Celsius. Bacteria operate around, 78, 77 degrees Celsius. And we can see that their optimal ranges are different. And then once they get outside of their optimal ranges, you can see that they have gone through a denaturing impact and they no longer are functional. Same thing, we know that the stomach is very acidic, so it makes sense for any enzyme that functions in the stomach to have a low pH optimal range, whereas the intestine is going to be a little more neutral or kind of on the low basic end of things. So we can see that the optimal pH for an intestinal enzyme, trypsin, is going to be around 8.0. So the last piece on enzymes, which are kind of cool, is that there are other factors that can come in and also play a part in the overall effect of enzymes. So those of us that go through and utilize vitamins, those are going to help aid in certain processes, and we might take like probiotics and that sort of thing. Those different ions and vitamins are going to affect enzymes and how they function. And most commonly, they're going to help increase the function of enzymes. So cofactors are going to be really small molecules that can go to the enzyme and bind to it. And they're going to be necessary for the enzyme to function. So iron, potassium, calcium, the enzyme isn't going to start working until those things are there. So sometimes if we're low in iron, we have to take some sort of supplement to boost it so that everything functions properly. your enzyme might be waiting for iron to come and attach itself, and then its active site will take its proper shape, and then the substrate can come in and do its thing. Coenzymes are going to be similar to those vitamin situations, where they're going to allow them to bind and again help increase activity. So if you're trying to figure out what's the difference between a cofactor and a coenzyme, cofactors don't necessarily have to be organic. So that would be one thing that might change. Or you can look at metal ions, anything like that are cofactors. And then our coenzymes are typically associated with vitamins. The last kind of piece that goes along with enzymes is there are other factors that can bind to them to stop their function. And so there are different inhibitors, which hopefully you see the word inhibit and you already understand that that means to stop or slow down or disrupt the overall function. So there's competitive inhibitors. If you're competing against something, you're trying to achieve the same goal, right? So competitive inhibitors are going to compete for the active site. Competitive inhibitors will come and lock into place at that active site where the substrate normally would go, and the competitive inhibitor is already here. So the substrate's going to slam into the competitive inhibitor and say, hey, I can't get in here, how rude, whatever. But that active site is not available. Non-competitive inhibitors are going to bind to a site called the allosteric site, a site that is not the active site. And it might be at the bottom of the enzyme, or it might be right next to the active site. But non-competitive inhibitors, right, if you're not competing for the same goal, but you are still competing against somebody, right, you might have two different end games. Substrates are still trying to get in the active site. The non-competitive inhibitor is trying to get to the allosteric site nearby. And once that non-competitive inhibitor binds here, it's going to change the shape and almost denature the enzyme so that, again, our substrates cannot attach and prevent that enzyme action. A couple more pieces. Again, in our POGL, we're talking about what does it look like for a couple different graphs. And if we increase the overall enzyme concentration out there, then we're going to see an increase in chemical reaction activity, right? That's going to be an increasing line, right? The more enzymes present, the faster the reaction can go and the more reactants can break down to products. And that's ever increasing, then it'll just keep on going faster. If we're talking about substrate concentration or the starting molecules that are out there, if we increase substrate concentration, we're going to see an increase of the chemical reaction until we reach a certain point, right? So at some point, all the enzymes are going to be locked and loaded with their substrates, and there are no more available enzymes to go through and continue those chemical reactions at that time. So when substrates equal enzymes, we're going to reach this point here, and this is called a point of saturation, or when all the enzymes are occupied, right? And so then we can see that it'll plateau or level off, and we'll have this even reaction rate from there on out. We'll go through this a little bit more later, but feedback inhibition is is going to be very similar to our cell communication topic that we talked about before, where we're going to see how certain enzymes can create inhibitors, or they can create coenzymes or whatever that might affect the overall chemical reaction, or they might affect the overall cellular communication chain. So over here, if we look at it, we have this initial substrate enzyme complex over here where threonine attaches to the active site, right? We can see that there is also an allosteric site over here at the bottom of the enzyme that's unoccupied. So the enzyme is functioning, right? And they can communicate and go through and say, hey, I'm enzyme number 1, we need to go through this feedback loop so that all the neighboring enzymes are also activating at this time. So it'll communicate, and enzyme 2 will function and create a product called intermediate B, which will attach to enzyme 3, which will create a product, and intermediate C that attaches to enzyme 4, so on and so forth, until we have our end product, or our final piece of information that comes from our cellular communication, right? Once that end product is released, this end product could go back through and be a molecule that shuts down our overall feedback loop. So if we have enough of what we need, this end product can come back through and attach at the allosteric site of that original enzyme to shut down the entire feedback loop, right? So here it is attaching at the allosteric site, changing the conformational form of our enzyme, and therefore it can no longer be active to the original products, and this entire chain would stop. That's called feedback inhibition when it stops. And then sometimes this end product might be something that helps as like a coenzyme or a cofactor that can go through and assist in other feedback loops. So there you have it.